Okay, we're on to translation, which is the second step of protein synthesis. We got into this a little bit in the last video, and we'll continue it right now. We went through these already, but translation, they really named it appropriately. It's going from the language of nucleic acids to the language of amino acids. So we're taking an mRNA message, remember that's just a copy of a message of a gene from the nucleus, and making it into a chain of amino acids that we can call polypeptide chain. We just remember a couple basics here. Proteins are made of subunits called amino acids. There are 20 different amino acids, and we put those 20 different amino acids in different amounts and different combinations. They'll make all the proteins that make the phenotypes of organisms. So from a bacteria all the way up to us, the phenotypes are proteins. And just remember the word polypeptide, we're saying an immature protein. It has to go through the rough ER and the Golgi apparatus to be folded in the correct shape and then be sent to the proper destination so it can actually be called a protein. Okay, I believe we went through this as well. We really need our three types of RNA to make translation happen. The top one there is the mRNA, stands for the messenger RNA. Remember that carries the DNA message out of the nucleus into the cytoplasm, because the DNA is in the nucleus, and that's where the blueprint is for this genetic code. But you make the proteins out in the cytoplasm, so you need that middleman, and that middleman is the messenger RNA. Take the DNA code from the nucleus, bring it out to the cytoplasm. We also need probably hundreds, if not thousands, of tRNA molecules. There's about 20 different types. Actually, probably a little more than that, but there's 20 different amino acids that are found on the different tRNAs, and they're floating around in the cytoplasm. And when the third thing there, the ribosome, calls for a certain tRNA, it matches up to the messenger RNA, brings the amino acid over, which then becomes part of the polypeptide chain. The ribosome is made of our third type of RNA, and that's called ribosomal RNA. We said in the last video that the ribosome is not surrounded by a membrane like the membrane-bound organelles are. It's protein along with ribosomal RNA. Alright, let's take a look at this next. A couple more basics. When the mRNA is read by the ribosome, it's not read one at a time, it's read 3 nucleotides at a time. 3 nucleotides together is called a codon. It also has the word code right in it. So the code of the mRNA is read in 3s. And remember, RNA is just made of A's, U's, G's, and C's. So 3 of those together in order is a codon. One codon will specify one amino acid to go into that protein that's being made. So if we take a look at this sequence, UCGAUUGGC, that would be read by the ribosome as 3 different codons. It would read UCG and then bring the right amino acid that would match up with that. Then it would go over to the AUU and read that. And if you look down below there, AUU is the code for an amino acid called isoleucine. And then it would read GGC. GGC is the code to call for an amino acid called glycine. So you have to understand that the mRNA is read in 3s. 3 nucleotides is called a codon. Each codon specifies a specific amino acid. So let's go back up to this picture here for a minute. This is a tRNA. Remember, the T stands for transfer. A tRNA, its job is to bring one of these, this is the amino acid right here, bring an amino acid over to the ribosome when it's called for. Now, how are they called for? Well, look down here. This CUC, this is on the mRNA. Well, when the ribosome reads CUC, it calls for an mRNA that has this portion here. This right here is called the anticodon loop. So remember, this down here is called the codon on the mRNA. It matches up to a very specific anticodon on the tRNA. So we just follow the base pairing rules here. A C would match up to a G, a U, remember there's no T's in RNA, would match up to an A, and a C would match up to a G. That's how this codon calls for this specific anticodon, again called the anticodon loop, which is then at the other end has this amino acid attachment site, and here it's leucine. So that's how it all works. That's how the ribosome can read a codon, call for one specific tRNA that has one specific amino acid on it. And we'll have lots of practice with this as we practice all of these ideas. Just make sure you write down everything over there on the side. Okay, so back to translation. The mature mRNA leaves the nucleus, has all those post-transcriptional modifications done to it, where the introns get cut out, the exons get spliced together. It's now the mature mRNA leaves the nucleus through a nuclear pore, comes out into the cytoplasm. The ribosome needs to know where to start on the mRNA. Just like in transcription, the RNA polymerase needed to know exactly where to start. Well, here it's called the start codon. The ribosome binds the mRNA at a certain codon called the start codon. In every single mRNA, for every single creature that's been studied, so every single mRNA that's ever been studied in people, or in fruit flies, or in bacteria, or in plants, the start codon is always an A with a U with a G. The ribosome binding to the mRNA at the start codon is called initiation. Let's take a look at this picture. It looks very busy, but it's not too bad once you look at it. This little golf ball over here is the nucleus. All these little holes are nuclear pores. Here's our mature mRNA coming out. We know it's RNA because it's only single-stranded, and there's no T's. There's U's. And so they're taking the same mRNA, and they're just kind of redrawn it down here for us. And here's initiation. This big blue blob is our ribosome, and it's binding right where this AUG is. That's the start codon. And what it does, A's match with U's, U's match with A's, G's match with C's. So this codon will only match an anticodon of a tRNA that's a UAC. The ribosome finds a tRNA with UAC floating around, brings it over, and plugs it on right here. These will form hydrogen bonds due to the base pairing, just like in DNA. At the amino acid attachment site of this is a methionine. That's the amino acid. That's called initiation. The next step, the ribosome reads the next codon, which is UUC. What goes with the UUC? That's AAG. So in AAG, the ribosome facilitates that bonding, and that has a phenylalanine amino acid on it. What the ribosome will do is it'll make a peptide bond between these two amino acids, just like that. This tRNA actually goes away. It's done its job, and the ribosome is going to shift over one codon. So this big blue blob leaves the UAC, and it reads the AAA. What goes with AAA? UUU. So this will dock. The ribosome will make the peptide bond between this phenylalanine and the lysine right here. And we're already starting to make a polypeptide. We made a 3 amino acid polypeptide chain here. Methionine with a phenylalanine with a lysine. And this just keeps going like this. And we're going to talk about when it knows to stop in just a minute. So that step that we just talked about after initiation, after the ribosome is docked with the AUG, is called elongation. The mRNA gets pulled through the ribosome. The ribosome reads a codon, matches a tRNA, makes the peptide bond, reads the next codon. So that's what this says here. As the ribosome reads each codon, it pairs it with the corresponding tRNA. That step is called elongation. Ribosome just keeps reading 3 at a time, 3 at a time, 3 at a time, bringing in those amino acids, dehydration synthesis, making the peptide bonds. The old tRNA goes away, it goes to the next codon, brings in the next tRNA, dehydration synthesis makes the peptide bond. The old tRNA goes away, and it just keeps happening. It's very almost like a machine working. Okay, we've said all this. And let's focus on this for a minute. The ribosome forms peptide bonds between the amino acids in the protein chain. And how does translation know when to stop? It stops when the ribosome reaches something called a stop codon. The stop codon is just like the terminator sequence in an in the DNA strand. It tells transcription to tell RNA polymerase where to stop. The stop codon tells the ribosome where to stop. Once the ribosome reads the stop codon, there's 3 of them. You don't need to memorize them. But there's 3 stop codons. That step is called termination. At that point, the ribosome breaks off from the RNA strand. It can go to a different RNA strand and make another protein. And that polypeptide chain, which is amino acids held together with peptide bonds, now goes to the next few destinations in the cell. It now goes to the protein. We should probably still call it a polypeptide. Next goes to the rough endoplasmic reticulum, which folds it into shape. And that's the opposite of denaturing, folding into the correct shape. It'll then go to the Golgi apparatus, which will label it, modify it, finish it off, and then it then goes either wherever it's supposed to go, in the cell, into the membrane, outside of the cell, and goes and does its job. So let's just come back to this picture. So this is what we're looking at here. And so this process would just keep going on and on and on again until eventually, let's see, 1, 2, 3, 4, 5, 6. Let's say this was the stop codon right there. Once it reaches this stop codon, the stop codon does not bring in a tRNA. What will happen is once the ribosome reads this, the ribosome breaks away. Any tRNAs that are still there leave, and this polypeptide chain then is directed to the rough endoplasmic reticulum. It seems like a lot. There are crash course videos on protein synthesis. There are Bozeman videos about this. There's a lot of YouTube videos that just show you animations of this. We'll practice this a lot. I have a few different activities we'll do to practice this, to work through this, to see how all this works. It'll make sense the more we do it. So if you feel a little overwhelmed with this, it's okay. It's the first time you're learning it. It's a very abstract kind of concept. We'll keep practicing it, and it'll make a lot more sense to you.